Hi and Assalamualaikum warahmatullahi wabarakatuh. My name is Wanurul Afika binti Zainuddin. I'll be present the diversity of marine phototroph. This slide will show you the flow of understanding from traditional to current understanding of primary producer. So I'll be speak in Malay and English so that it will be easier for me and you to understand what I'm going to talk about. Okay, for the introduction of marine phototroph, we have to know what is marine phototroph. Marine phototroph are aquatic living organisms that require light energy to produce ATP either from carbon dioxide or any other substance. In this topic, there are 5 terms that commonly used. For number 1, photosynthesis, which is a process that requires sunlight to produce organic compound from inorganic carbon, such as sugar. Number 2, phototroph. Phototroph is organism that requires sunlight to produce ATP. Number 3 is photosynthetic organism. Photosynthetic organism is organism that use photosynthesis to produce energy. And number 4 is oxygenic phototroph. Oxygenic phototroph is a phototroph that release oxygen from their photosynthesis process. So the last one is primary producer. Primary producer is quite similar to phototroph, which is organism that can produce ATP. But primary producer can produce ATP in presence of sunlight or no sunlight. And they can produce organic compound from inorganic carbon. Next, this slide shows you the timeline of diversity marine phototroph. In the year 1970s, traditional understanding stated that primary producers are mainly performed by diatoms and flagellates. But in the next years, 1980s, 1996, 2000s, 2007, 2010, and 2011 showed that many marine phototrophs are yet to discovered. This is automatically change the traditional understandings. One point one, traditional understandings of primary producers in 1970s. In the year 1970s, they still believe that the main primary producers are diatoms and flagellates. This is because the lack of advanced instrument prevents them to discover phytoplankton that smaller than 60 micrometer. In the year 1970s, they only have plankton net that with mesh size of 100 micrometer, 70 micrometer, and 60 micrometer. But in the late 1970s, with the help of fluorescence microscopy and collection knowledge from electron microscopy, a cyanobacteria was discovered with called Cyanococcus spp. Cyanococcus spp is about 0.5 to 2.0 micrometer in length and 0.5 to 1.0 micrometer in diameter. Next, 1.2, the discovery of divinyl chlorophyll A in 1980s. By this year, the instrument that can identify compounds as tiny as part per trillion is called high performance liquid chromatography and cell detection techniques by using flow cytometry. By these instruments, DV chlorophyll A was discovered. DV chlorophyll A is slightly different from chlorophyll A, which is it had different in spectrum absorption. DV chlorophyll A also presents in a cyanobacterium called Prochlorococcus, and it was used in oxygenic photosynthesis. DV chlorophyll A also efficiently absorbs blue light in the ocean. And currently, you know what? Prochlorococcus and Cyanococcus spp are the main primary producers with contribution of 70%. They are also dominant in tropical and subtropical. So we continue in 1.3, the discovery of oxygenic photosynthesis by far-red light in 1996. Chlorophyll D is a pigment that can absorb far-red light about 30 nanometer wavelength longer than common chlorophyll A does. So, this chlorophyll D is present in Acariochloris marina cyanobacteria. It allows its cyanobacteria to undergo photosynthesis even in highly filtered light environment, which means in a situation that limited visible light, the cyanobacteria still can undergo photosynthesis to produce their own energy. Now we move to the point 1.4, which is discovery of photoheterotroph in the yearly of 2000s. There are two discovery of photoheterotroph, which is the first one is in the yearly of 2000s. Bacterial chlorophyll A was found in the aerobic and oxygenic photosynthetic bacteria AAPB, such as Erythrobacter. B chlorophyll A is a common pigment in anaerobic and oxygenic photosynthetic bacteria NAPB. This B chlorophyll A can be found in non-sulfur purple bacteria. Its peak absorption of spectrum is in vivo of 800 to 805 nanometer and around 870 nanometer. For the second discovery is a bacteria that contains proteorhodopsin, which is the proteorhodopsin is a light-dependent proton that can supply energy to microbial metabolism. These both discovered bacteria are photoheterotroph, which not relying on carbon dioxide as sole carbon source to produce organic compound. It can also be alcohol, fatty acid, and carbohydrates, which is organic compounds as their carbon requirement. By the year 2007, the unknown phyla of algae was found. An instrument with ability to define direct genetic analysis, which called metagenomic analysis, was used to detect the existence of the algae which has not belonged to any phyla. This algae was called picobeliphites. Through some research, picobeliphites is at least consists 3 different clades, which the 2 of them has been recognized, while the other was not. 1.6 Chlorophyll F in 2010. Chlorophyll F is another redshifted chlorophyll and the longest wavelength chlorophyll that has been found. Its maximum spectrum absorption is around 700 to 20 nanometer and in vivo 750 nanometer for far red light. In 2010, the studies of chlorophyll F did not end with enough knowledge of its contribution in the marine phototroph, but some researchers continued these studies and found a cyanobacterium that grown under 750 nanometer far red light. It consists 90% of chlorophyll A, 10% of chlorophyll F, and less than 1% of chlorophyll D. The cyanobacteria called as Crococcydiopsis thermalis. Lastly, in the year 2011, Repomonads was discovered. By using metagenomic analysis targeting plastic DNA, the new phylum was discovered. The new phylum was tentatively named as Repomonads. Their habitats are both in freshwater and seawater. Some researchers also took water sample from Sargasso Sea near Bermuda in late winter and obtained abundance of Repomonads. As for the conclusion, the first understanding for primary producers was mainly performed by diatoms and flagellates. That is because the lack of advanced technologies. As time passed by, many improved instruments were developed to help the study of marine environment, such as high-performance liquid chromatography, cell detection techniques by using flow cytometry, and metagenomic analysis. So, many other types of phototroph were discovered and changed the traditional understanding on primary producers. So this is the end of the slide. So I hope that you can understand what I was explained, and thank you for lending your ears.